Okay, there's 1 more minute. We're going to start exactly at 4. Um, all of this is going to be recorded in YouTube, and I uh, would like to announce that this basic colloquium, which is on Nobel Prizes, is the highest representation of science that is being awarded in all the fields. And I hope this institute is alive in discussing science. And I hope faculty members come up more in colloquia as well as bring their students. It's a fervent appeal to all faculty members right in this institute to bring their students. If we are not excited about Nobel Prizes, we should stop doing science. Please come. Okay. It's actually a great pleasure to organize this colloquium once again. Every year we do this with which is celebration of the 3 prizes that are declared every year from the Royal Swedish Academy. And this year has been sort of a very intriguing set of prizes that have been sort of awarded, because the things that have happened in last 10 years have been rewarded in a very quick pace. So we will hear the description of how our world is changing with machine learning, artificial intelligence, and neural networks, along with um, fields which are traditionally, of course, experimental centric, getting a prize for understanding how biological networks work. So um, I would like most of you to come in front if those seats are filled in. And we will start with our first speaker, Professor Ullas Kolthur, who has come all the way from Hyderabad to give this in-person colloquium. And he is always enthusiastic about discussing the work that goes on in his lab, of course, through colloquium series as well as through this. So Ullas, thank you for doing this, showing this enthusiasm. And we'll start with you with the Nobel Prize in Physiology and Medicine.
Okay. Okay, great. Thanks, JD. I don't think I need a reason to come to Kolaba. I still belong to Kolaba. Don't make me an outsider yet. Um, so, um, so the Nobel Prize for Physiology, unlike what JD said for the other two streams that we're going to be discussing today, has not been such a recent discovery. Actually, this has been in about 3 decades now. So the other two were, more recent. And of course, it also highlights how biology is ruling the entire science now, because everything has some biology. All the physicists who are still convinced that biologists are lesser mortals, please rethink. Okay. So this is an interesting prize, and it was waiting to happen because it changed the way we think about how biology operates, particularly something that is so quintessential biology in terms of how your genetic material gets expressed, and that expression is what leads to pretty much everything that happens in biology. It doesn't matter whether you are an archaea or a bacteria or a human being. So the Nobel Prize for Physiology and Medicine goes for the discovery of microRNA and its role in post-transcriptional gene regulation. I think it says it all, but if you still want to know more about what post-transcriptional gene expression is, we'll go through a little bit of details. So this is the citation officially that is released by the Nobel Committee. And the two people we are talking about are Victor Ambrose and Gary Ruvkun. So I've just highlighted the key elements that I think are going to be defining for why the Nobel Prize was given. Of course, the other details are there. So basically, they discovered microRNA, which is a new class of tiny RNAs, and we'll see what those tiny RNAs are, which changed the way we think about how gene expression regulation happens. They came up with a new principle of how gene expression regulation happens. And this added a new dimension to the entire biology. And in fact, now it's all pervasive. In fact, particularly for higher eukaryotes, you can't think about any gene expression without thinking about microRNAs. Now, for the people who may not have had enough exposure to biology, all of us know that genetic material is mostly DNA. Of course, there are organisms that also contain RNA as their genetic material. Ultimately, it has to get converted to DNA for, again, making proteins. So what you have is a genetic material that gets expressed as what is called a messenger RNA, called mRNA, which gets translated through a machinery called ribosome, which also got a Nobel Prize some time ago. And these proteins are the ones that pretty much make up everything that we think about in terms of what functionality is achieved from a biological perspective. Now, biologists have always been wondering about the complexity of the organisms, not just in terms of size, shape, and the different types of cells that exist in human bodies, but also in terms of complex functions that higher eukaryotes have. If you actually think about the number of genes that we have from a lower organism, like, let's say, C. elegans, which got the Nobel Prize this year, I mean, in the sense the microRNAs were discovered in this worm called C. elegans. If you look at the number of genes have not been very dramatically different. There's a non-linear increase in terms of the complexity that you see in mammalian systems. So largely that has been contributed because of a small subset of genes, which is about 25,000, giving rise to more than a million kinds of proteins, which means that you have a huge repertoire of proteins which perform very different functions. But very little has been actually understood about spatial and temporal complexity of expression. That means you can create number of variables in terms of the parameters that you can have, but how do these parameters come about in space and time has been a major challenge for biologists. And this is where, again, this particular discovery has kind of a groundbreaking feature. But I'll step back and I'll introduce a little bit about what was happening parallelly in the field and why these people were even probably thinking about post-transcriptional regulation of gene expression. So, of course, we know that the history of who deduced the structure of the DNA, and one of the bulky. How many of you don't know what an RNA is, so that I can tell you what an RNA is quickly? Really, Amartya? Okay. So RNA is a ribonucleic acid, which is what is one of the nucleic acids that are present in the cell. You have DNA as the genetic material. RNA is the intermediate between DNA and protein. But RNA was the more prebiotic or primitive macromolecule. In fact, there is a concept called RNA world, and that's why you have a lot of RNA viruses that still exist, right? So RNA is a polymer like a DNA. So it is the one that carries the message from the nucleus to the cytosol, but they do many other functions. I'm going to elaborate a little bit more on that. Is that good enough, or should I go into more details? I think that should be okay, right? So you have a lot of RNA molecules that were also discovered. For example, this is called rRNA, like I said, ribosomes, where the mRNA, which is the messenger, gets translated into the protein, and that's what gives you the functional counterpart. So people were identifying all kinds of RNAs between after the DNA was identified and up until early 2000s. And one of the key discoveries was this fact about what are called interfering small micro RNAs that came about, called siRNAs. That means small interference RNAs that are largely present in the plants and also to some extent in lower eukaryotes themselves. So these are small RNAs which come and regulate or negatively regulate the expression of some of the host genes. And they were also evolved as a defense mechanisms against viruses. That means, for example, a plant gets infected by virus, your siRNA or dsRNA based mechanisms are the ones that actually comes up and inhibits the RNA replication of the virus, because the small RNA can negatively regulate the invader. So that's how this happened. So there are a lot of small RNAs that were discovered in this era, and this is something that, if people are interested, you can go back and look at this particular publication. Simultaneously, people were started to discover or sequence genomes across species, and of course, Human Genome Project itself was a huge effort, and what took almost nearly a decade, now you can sequence the genome in a couple of hours, actually, literally, right? Now, when the genome sequence was evolved, people realized that the number of genes that actually code for proteins are very small. Only 3% of your genes code for proteins, which make up your this 100,000 or tens of hundreds of thousands of proteins that give you the functionality. That means you have accumulated junk DNA. And in this particular paper, they equate it to dark matter and dark energy in cosmological terms, and says that, look, there is dark genome, and the dark genome is probably shaping the white genome or the fair genome, right? And the fair genome is the one that is giving you these mRNA and proteins. So you have a whole lot of other genetic elements or gene segments which are coding for something else, coding not as a protein but as several other RNA molecules, all of which are actually important for making this transition from DNA to mRNA to protein, but they also have several other functions, including tuning the functions of the proteins themselves. That means you have a huge repertoire of how things happen across the scales, right? So therefore, what happened was that while people were identifying this small RNAs that were negatively regulating gene expression, people also wondered about what was the rest of the genome contained, and simultaneously people put things two into together. And in fact, here I just wanted to you to appreciate that in humans you have a lot of genome that doesn't contribute to proteins, and I did tell you that the proteins are the one that gives you a lot of these functions that we have. Okay, fine. Now, what is the background that was led to this particular discovery, right? The background was that people, before the genome was available in terms of the sequence, and we all belong to an era where no fancy technique was available. You didn't even know the genome sequence, because you didn't know how to sequence large segments of DNA. You could only sequence small fragments of the DNA, right? So the best approach is to identify what genes contribute to the function was take what is called classical genetic approaches, in which case what is called forward genetic and reverse genetic approaches, and people in biology have given several colloquia on this. But one of the popular ways to do this is you mutate genes in the genome by multiple ways. One could also be what is called a directed mutagenesis, and something called also what is called random mutagenesis. That means you randomly mutate genes and you score for phenotype, or you can also do targeted mutagenesis. So that allowed people to ask what fragment of the DNA was responsible for the phenotype that you wanted to score for. For example, you wanted to look for a hair color, you mutate a particular gene, you now know which you could go back and map the gene through multiple ways, both genetic and molecular, and say, okay, maybe I've identified a gene that is responsible for color of eyes, color of hair, color of whatever, height, whatever you could think about. So that's the approach that people were taking, and C. elegans became one of the favorite models, like fruit flies, to do some of these genetic screens for obvious reasons, because they were easy to grow, they had a shorter lifespan, phenotypes were easy to score for, they were less complex when compared to humans, and of course, almost no ethical issues. Although you can kill as many flies as you want, and nobody's going to take offense to it, right? Anyway, so people were trying to look at the fundamental principles of how cells, give rise to organisms. In the sense, if you remember, all of our cells are born out of what is called a zygote, which is very small, and then you give rise to all the different types of multiple types of cells. So people were wondering how development happens and how different cell identities emerge during development. So a lot of work had happened in the past, and ultimately some of these papers led to the identification of several of these genetic loci or gene fragments or genes themselves, which led them to hypothesize what would their functions be, and all of these were involved in regulating the development or cell fate identities of the worms or other organisms that people thought about. So two of those were called Lin14 and Lin4. Let's not specifically worry about the names, but basically there is a 14 and a 4, and these two were the one that came out as one of the things that were important for regulating this developmental processes, and there are several of the cell biological processes that were important for development. Now, what's interesting is that I put this here. So there was this already a suspicion that what I said is that you have a lot of mRNA that is produced, right? So you have the genome, you have the DNA, and the DNA gives rise to RNA, and RNA gives rise to protein, right? This is very fundamental. It's almost like a God-given truth for biology, right? You have DNA, RNA, and protein. So usually, if the amount of protein has to be regulated, you need this to be regulated and this to be regulated. Usually, people assume that the amount of RNA is equal to the amount of protein, right? Because if you have X number of RNAs, you should have X number of proteins, right? Because you would have done the translation. But because the one mRNA can give rise to multiple proteins, there could be a scaling factor. You could say 1:10, let's say. But what was surprising is that people found that even when the mRNA stayed constant, these protein levels went up and down, right? That means somehow this amount of protein did not correlate with the expression of the RNA itself. There is a discrepancy in terms of the messenger amount and the final product. This could be either because this is getting degraded, of course this goes down, but it could also be possible that there was a regulation at the step of conversion from RNA to protein that was already kind of guessed or hypothesized by some of these papers that happened earlier. So if you're interested, these are very classical papers that have used beautiful old style biochemistry, molecular biology, and genetics, which have led to some of these identifications. So then ultimately what happened is that they identified this particular small RNA called Lin4 that negatively regulates this particular gene called Lin14, and all these microRNAs are actually kind of conserved across species, and now it is known in the context of humans. It is relevant for almost everything that you think about what happens in biology. Even your neural processes and your thinking and your behavior development, everything is dependent on that. So basically, the seminal papers that led to the prize themselves are these two papers titled here. So this is a real picture, this is a cartoon. Essentially, I'll go through these papers and tell you why this was so important, although there was evidence to say that one of the Lin4 was negatively regulating Lin14, and these two levels were kind of antagonistic. And there was already this understanding that the amount of RNA does not correspond to the amount of protein, and therefore there should be some regulation intermediary control mechanism that existed. So what was the first paper that came about? This was Gary Ravkin's. Of course, they were published back to back in the same issue. And in fact, each of them cite the other paper. That means it was probably a co-submission in current consultation with an editor, because otherwise, how will I know who else is sending the paper, right? So one of them cites the other paper and that person cites this paper. So that means they were already kind of worked it upon together. So what this gel says is that, remember, I told you there is this particular thing called Lin14 that negatively regulates Lin14, Lin4 that negatively regulates Lin14. That means if I have a worm that does not contain Lin14, this is basically a mutant. This is Lin4 expression, which is this is the protein expression or the mRNA expression. You can see that whenever I don't have this, expression of this increases. That means that tells you that there is a regulatory step in between, and that regulatory step is probably Lin4, right? And essentially, when they mapped the genome sequence by painful sequencing, they actually found that the sequence of this particular RNA and this particular RNA had some complementarity. That means they were able to base pair with each other. So that means there was some rule associated with how these could interact physically, and that physical interaction was necessary for this regulation about mRNA being controlled to see how much of protein can be made. I hope you're getting the point, right? Is it okay? Sure? Amartya? Okay, good. If you can get it, I think I'm happy. Yeah? Sorry, I didn't mean to dig. I just said because you didn't know RNA. He is the messenger. Okay, now that was the first paper from Gary Ravkin, right? What did Victor Ambrose did? What Victor Ambrose did was that he actually painfully mapped the genome locus that belonged to this particular Lin4, right? So as you know, the DNA is very long polymer. You have small segments that can be considered as genes, right? Now these genes normally, non-biologists think, all of them lead to protein. But now we know a lot of these contribute to RNA that can regulate how the protein is expressed and how protein functions itself. Now what they did is, using, again, like I said, very painful molecular biology experiments, they mapped this particular locus in the gene, and they called it as Lin4 locus. And what they identified is that they found that this was present in a segment of the DNA which could not encode any protein. That means, remember, they identified this as a gene. If this gene had to regulate this gene, popular perception would have been this also codes for a protein, and this protein is the one that regulates the other protein, right? But turns out that this fragment of the DNA did not encode any protein. That was a fundamental breakthrough that was provided by this particular paper. That means something that we thought will regulate this particular expression was not something fit the classical understanding of biology. It meant that there is some other molecule which regulated this, and that turned out to be an RNA. Later, he actually purified these RNAs and showed that the RNA that regulates this is a very short RNA, which is about only 22 to 23 nucleotides length. Now, typically mRNAs are very large, can go up to several hundreds of base pairs or thousands of base pairs, okay, or bases rather. There is no base pairing there. It can go to up thousands of bases, whereas these short RNAs are only between 22 to 23 nucleotides. So that means a very short RNA, that's why it's called microRNA, because very small in size. These microRNAs can ultimately regulate this, and he also showed how that this base pairing happens. That means there is this small 22 nucleotide RNA that is binding to some large RNA, and that is what is negatively regulating the expression. Clear? Yeah? Okay, fantastic. Now, I'll fast forward and I'll tell you what is the current status of the field. The current status of the field is that we know that actually all of these microRNAs also have to be transcribed. Transcription is a process where you go from DNA to RNA is called transcription, right? DNA to protein, RNA is called transcription. Anything that happens after RNA is called post-transcription. Simple English. So what happens is that these RNAs are made as transcripts. They are themselves processed, exported from the nucleus to the cytosol. In the cytosol, remember I told you these are 22 nucleotides. These mRNAs are several thousands of bases long, and these small RNAs can bind those big RNAs and can regulate the fate of these mRNA. And this is the mRNA that gives you the protein. So what you are saying is that a tiny factor that people would have ignored otherwise as fragments that are inconsequential are regulating the entire genome, because they can regulate the expression of multitudes of RNA that you can think about in biology. So now, how do they do this? Small microRNAs can bind to these larger mRNAs, and then they can either lead to degradation of this RNA. That means, suppose if I have a DNA that is leading to mRNA, this has a fate of giving rise to protein. Either it can degrade this small RNA can degrade this, right? MicroRNA can degrade this, or it can inhibit this and not make it into a protein. That means it was a clear regulatory mechanism that was happening, right? Now, so how does this function? This functions by this. You have one microRNA that can bind to several mRNAs, which is interesting. One mRNA can be bound by several microRNAs, right? How does this function? It's again a combinatorial nightmare. Very few people have taken theoretical approaches. There are some papers, but it's far fewer than what should have happened. So you have an mRNA which is very large, right? You can have several microRNAs that can bind them. That means there could be A, B, C, whatever, and each of them can have a different regulation of the mRNA. Each of these microRNAs can also bind several different mRNAs. For example, this could be a gene that codes for your hair color. This could be a gene that codes for your eye color. This could be a gene that codes for your skin's color, or whatever. So the same microRNA can bind to different genes and regulate. The same mRNA can be bound by different microRNAs and can be regulated. That means the combinatorial space increases vastly, and this combinatorial space actually what also drove evolution and what gave you the different functions that we all enjoy as human beings, with all complex behaviors and complex physiological systems. So since I'm running short of time, I will skip everything else, and I will tell you why it is also so relevant in terms of even diseases. Almost every known disease that you can think about have an underlying basis of these microRNAs going bad. Either these microRNAs are not expressed or they're overexpressed. If they're not expressed, the target mRNA will be hyperexpressed. This could be a disease-causing gene. On the contrary, you can have an increased expression of these microRNAs that leads to downregulation of the mRNA. That means you can, again, this could be a protective gene and it could lead to diseases. So microRNAs have emerged as predominant regulatory factors that are involved and implicated in almost every disease, from neurodevelopmental disorders to neurodegenerative disease to Alzheimer's, Parkinson's, all the way to cancer and obesity and diabetes. Therefore, it's not surprising to see that several companies have come up with what are called mRNA-based therapeutics. That means people are actually actively using miRNAs as therapeutic agents by simply injecting them into humans or mice. You can regulate the expression of some of these mRNAs. Now, some of these microRNAs have already been FDA approved. In fact, for hepatitis virus and several other viral diseases, microRNAs are already in clinical use. Several others are in phase 1 or phase 2 or phase 3 clinical trials. And we'll see a lot of these miRNAs coming up as drug targets or therapeutic agents to regulate these mRNAs and therefore give this. Now, what are the outstanding questions in this particular field? There could be several, but what I particularly like, what is outstanding is, now the genome, remember, because if I take a gene-centric view of biology, the genome is a long strand of DNA, right? All of us know this. This long strand of DNA doesn't exist as a naked DNA, right? This doesn't exist as a naked DNA. This is actually wrapped around by proteins that gives you conformations that are unimaginably complex. People have not even thought about how this happens. In fact, there is an entire field called epigenetics that actually says you can regulate the expressions of genes by not changing the sequence of the DNA, but changing which gene gets expressed when or how. That means I can have the same gene, all of us have the same gene, but you might have a different phenotype than I do because my genes may not be expressed because these proteins can either repress the expression of genes or activate the expression of genes. Now, this genome is looped into multiple conformations as I've shown here, and these are really higher order architectural loopings in the genome. We still don't understand what regulates these architectural loopings, and these architectural groupings are the ones that provide some kind of a language for proteins to be expressed. If protein coding genes can be present in these architecture complexities, can microRNA genes can also be present in architectural complexities. That means there is a state and a function which is regulated by expression. The other thing is homeostasis of microRNAs is very poorly understood. We know some, but we don't know many things about how do cells maintain X amount of, let's say I have an microRNA concentration. How do I maintain this particular mRNA concentration, right? What is exciting is that some of these miRNAs can be exported out of one tissue. For example, liver can export these microRNAs to muscles, and there is a cross-tissue communication. They can actually act as hormones and regulate one tissue or the other. That means the complexity becomes even crazier or greater because microRNA of one tissue, for example, brain microRNA can regulate muscles, or muscle microRNA can regulate brain. All kinds of combinations exist. For physicists, if you are interested, thermodynamics of this is not clear. Thresholding is completely not understood. We have not really approached anywhere close to systems level interplay of microRNAs at a multi-organism level. Thank you.
We will take a few questions and then we move on to the physics prize. But yeah, questions. 14. Yeah, ask.
Hi, Ullas. Very interesting that. So you initially mentioned about siRNAs, right? Small interfering RNA or RNA interference. I believe that also got Nobel Prize much earlier, right?
Yeah.
So how does microRNA is actually different from siRNA in the whole biology setting? And then why actually microRNA actually the prize was really delayed because the research has been going on for decades, right?
Correct. So why it is delayed could be many reasons, including politics. So let's not go there, right? So the small interference RNA is very different. Like I said, it is restricted to a specific kingdom or phyla, right? Kingdoms of phyla. Whereas microRNAs are all pervasive in some sense. It's present in plants, it's present in humans, it's present in pretty much everywhere that you can think about. The mode of mechanism itself is different. Usually, siRNAs always lead to degradation of the mRNA. MicroRNAs never lead to degradation of mRNA. You can actually, so the beauty of biology is that suppose if you're going through some oscillation in your physiological state, where you need a constantly high amount of protein that should oscillate, I don't have to make mRNA degrade it, make mRNA degrade it, make mRNA degrade it. I can make mRNA, keep it, I can translate, not translate. It's almost like applying a brake and not applying a brake. You don't have to dismantle the entire engine. right? So what microRNAs do is this applying the brake kind of a mechanism, which is very different from what an siRNA does.
Thank you.
Yeah. Hello. So my question is that, as per I understood, this microRNA is generated from the parent main mRNA or
not necessary. They
Not
can be
necessary.
generated from the parent mRNA. They can be independently expressed from a totally different locus of the genome.
Okay, so is there any mapping to what cellular conditions are leading to whether overexpression of microRNA or?
Yeah. So there are a lot of studies. For example, we also do a lot of work on microRNAs. In fact, we show anticipatory expression of these microRNAs. That means if these microRNAs are required at another stage in your day-to-day life, it is expressed as anticipation. They're kept unprocessed and they're processed when they're required. So you have a lot of these regulation that is happening.
Okay, thanks.
So I have a question about the evolution of microRNAs themselves, because for microRNAs to function, they must be complementary to the mRNA to a certain extent, right? So which means that, where did they actually come from? Are they pieces of mRNA which then got, transposed to some other location, which then later became microRNAs, or what is the field actually thinking about?
No, so the transposition theory doesn't fit well for the microRNAs. People think that errors in replication, it probably leads to slippages and copy number variations. Those kind of things could have led to these microRNAs. Because when the genome gets replicated, you have copy number variations that can go haywire in the genome, right? And then copy number variation can happen this way. The other way is, there's another concept called exonization. I don't know if you know this. ALU elements, okay, can get exonized. ALU elements exonization could also lead to generation of microRNA genes. Now, these are still theories. There's no way to prove it one way or the other, right? So in terms of evolution itself, yeah, it's quite likely it's not a classical retrotransposition event that could have led to microRNA generation. It could be either copy number variations, that means genome during replication, there could be errors in some fragment being over-replicated. So that could have led to this. Or one of the examples is ALU-based exonization that could have led to this. Yeah.
Okay, well, let's thank Ullas once again for a wonderful presentation on the idea. The next speaker, and this is actually in my tenure, this is the first time that I am bringing a person out of physics, not from a physics background, and just shows the power of the Nobel Prize this year, that things from in mathematics and computer science can be borrowed in to solve an important scientific question. And I think this is a primary example that we should, as schools of computer science and mathematics, we should talk to each other more. So Piyush Srivastava will be speaking about the Nobel Prize in physics.
Yeah, sure. Thank you. Okay. So thanks. So let me start. So this is going to be about learning with networks. There's going to be nothing in the talk which someone with some calculus and class 8 to 10 mathematics cannot understand. Nothing very advanced. And there's something that I found a bit jarring when the previous talk started, that it was said that everything in this prize was about 10 years ago. Actually, everything I'll be talking about will be at least 15 years old, and most of it will be 100 years old or 60 years old and so on. And so let's start. So here is what the citation says. It says, the Nobel Prize in Physics 2024 is given for foundational discoveries and inventions that enable machine learning with artificial neural networks. It's given to two people, John Hoffield and Geoffrey Hinton. And that's where the title comes from. What I will be highlighting is that this is a story of ideas that are simple in hindsight, and very importantly, they are so important, and so a measure of their importance is they have been rediscovered many times by different people in different contexts and used again and again in different situations. So this is not Michelangelo's painting. This is human creative genius, but this is not like the Sistine Chapel or Michelangelo's painting. This is more like Kailash at Verul. It is a work of several people over several decades in several fields, in several different contexts, which has combined and gotten us where we are. So there was another thing that was said about being excited about prizes. That also I found jarring. We should not be excited about prizes. We should be excited about the results. The results were there. Just because 10 people in Stockholm think it's interesting does not mean that it became interesting right now. It was interesting even then. So what we should think of it as an opportunity to maybe revisit the history of the subject and see what has happened. So this is what I will do. This borrows heavily from a particular survey that I cite several times, actually 2 of them. So those citations will come. Okay, so let's start. And so I don't have a physics background, but I have seen or I have read some of these Feynman things. So you should start always with magnets. So everyone is excited about magnets. And this is also about magnets. So magnets, everyone has seen them. There are some nice properties. They have memory. This is something very important. It's probably the first thing we see in our physics curriculum in school that has memory. It has a certain state. It has north-south polarity. And this memory is not just a memory in the sense that, okay, it's there. It can be removed. You hit, I mean, this was one of the experiments you did, right? You take a magnet, put it on the ground and hit it hard. Magnet may disappear if you are lucky. And of course, if you heat it enough, it goes away. So it can be altered. So there is memory, it can be altered. And this is important. That's how our computer memories used to work till about 5-10 years ago. Actually, that also won a Nobel Prize, I think, one of the. And the important thing that is about the memory, yes, it can be altered, but also it's important that it is stable to small perturbations. Otherwise, it will not be useful. Right? I mean, things, if your hard disk was losing data every minute, it would not be useful. So good. So this is what a magnet is. Funnily enough, people were trying to make toy models of magnets in statistical mechanics 100 years ago, and they were not succeeding. So Lenz created this so-called Ising model. So Lenz was the advisor, I think, and Ising was a student. Ising solved the model in 1 dimension. That's usually done as an exercise now. What is the Ising model? So it's like you model the magnet as some kind of collection of these vertices. Oh. Yeah, these vertices. So these are either in 2 states, plus or minus, and there are connections between these vertices. And the whole system has an energy. And what is this energy? There's some interaction term between pairs of vertices. And the energy term is basically like, if the interaction term is positive, then you basically, it just depends upon whether the 2 things agree or not and what the sign of the interaction. That's all that is. But it looks, there's some linear structure to it. Basically, if I know my own so-called spin, that's what it's called, that's sigma, then I just need to compute some kind of a sum over all my neighbors, and then I know what my energy is, or what my contribution to the total energy is. Okay, so that's what the Ising model is. So why were people failing? So what they wanted to recover is this effect that there is memory can be lost, and it is still stable to small perturbations. Ising essentially showed that there is no memory in 1 dimension. Okay, so there's no memory in 1 dimension. Okay, so this is often written as no phase transitions in 1 dimension. And actually, this caused a bit of a crisis in the sense that there is a story I've read about a conference in the 1930s called Van der Waals conference, where there's a vote taken that will ever these kind of models be able to reproduce phase transitions, and it narrowly passed. People, and this is supposed to be surprising because around the same time, Piles had actually shown more or less that in 2 dimensions, there is memory, although it was not sort of a very sharp transition. And Onsager showed in 45 that this is there. Okay, so why am I talking about magnets? We are supposed to talk about deep learning. So around the same time, so this is 1940s, around the same time, in a different field, the same idea was going to play a role. And this idea was that well-connected toy models of magnets do have memory. Well-connected is important, 2 dimensions or more have memory. Okay, so what were they trying to do? So this is Mekulo and Pitts, that is hidden, but it's like 1943. And one of them, I think, was a neuroscientist, and the other one was a logician. And this is a paper from a journal called Bulletin of Mathematical Biophysics, which I find quite impressive that such a thing existed in 1943. But and here is cut out from their paper, and they say that the activity of the neuron is an all or one process. Okay, that's first thing. And let's look at the second point. A certain fixed number of synapses must be excited within the period of latent addition. That just means within a period, in order to excite a neuron at any time. And this number is independent of previous activity and position of the neuron. So, okay, so what all that is being said is that the neuron at this time just looks at how many of its neighboring neurons are maybe active. That's their model. And then depending upon that, it excites or it is in excited state or not. So this is a very simplified model, but this is what they had. Okay. So here is what that model looks like mathematically. Yeah, I'm sorry, I have to juggle this, but maybe I'll do whatever. Let's see. So a slight generalization of this 0.2 basically says, let's put weights also. So it's just not sums. And maybe things start looking similar. So each neuron fires if the sign of whatever the weighted excitations of its neighbors is of a certain type. It's negative. Okay. And this is, many of people might have seen, this is the famous perceptron. We'll see what that is. But just remember this name, perceptron. But it also looks very much like the easing. Okay. So, but let's see what this perceptron is. So this perceptron is just this. It's like a linear function, something that does sums things linearly, and then thresholds. If they're above a threshold, it fires. Otherwise, it does not. Or let's say it fires at a plus level or at a minus level. So also, I don't know the history of this so well, but it does look to me that some of these things were also inspired by developments in electronics that were happening at the time. This idea of having thresholds which could do thinking probably did drive some of this work, but that's not so important. That's just to keep in mind. And what we want to do is the following. So this thing we want to say can learn. So what does it mean to learn? So maybe someone comes and classifies all patterns for us. So there are all these possible patterns, right? So this sigma 1 to sigma k, they can be all possible 2 to the k possible patterns of signs. For each such pattern, someone says, okay, these are the patterns that are yes, these are the patterns that are no. Of course, this looks arbitrary, but maybe this thing looks like, maybe if there are 10 plus, at least 10 ones, then you should say yes, otherwise you should say no. So that's the kind of thing. But what we want to do is that, of course, if someone comes to me and says, or comes to any one of us and says, oh, I want to train this perceptron to recognize all those sigmas which have at least 10 ones, it's very easy, right? What will you do? Okay, good exercise. So just set J in some way so that you can do it. But it's fine. But what we want to do is to have some general thing. We want to say that, okay, here is an algorithm. It will, if at all there's a solution, it will find it. And it should be a simple algorithm. It should be an algorithm that at least in some dreamlike state, one can believe that is what neurons or some organisms are actually following. Okay, so, and this is the so-called perceptron algorithm, and it's just written in one line. That's the algorithm. As long as there is some sigma for which your algorithm is wrong, just move J slightly. Slightly is important. Slightly, so towards classifying it correctly. Okay, so I'm not writing down the details. I will actually show you how this thing looks like when it's working. And in hindsight, the perceptron already anticipated two of the most important rules of thumb of modern deep learning. And these are both about being slow. So the first is to consume data slowly. You look at one input at a time and see, okay, here is this one thing I'm misclassifying. Let me correct things slowly. So consume data slowly, consume all of it, but slowly. Second one is to avoid premature optimization. Don't just say, okay, here's this new sigma, I have to classify it correctly. No, you just move a little bit. Maybe that doesn't help with this sigma right now, but you don't commit to getting this sigma right now. Okay? This example right now. So, okay, so how does this perceptron look? So here is how it looks like. Some of you might have seen this if you took this class, many of us taught. So you can see the date, and the date probably shows when this video was made. It was made for the class. So here is what, so this line is what that thing that is trying to learn, and those crosses are the examples it is checking. So when the example is correct, it doesn't do anything. But when the example is, let me just speed it up. When the example is bad, it moves. And sometimes, so we will see what starts happening. So it's, after a little bit, it is left with just that one orange point. So what it's trying to do is to separate the blue points from the orange and the green ones. So this is actually one of the most famous data sets, the iris data set, 3 species of flowers with just 4 parameters. And yeah, and you can see that it sort of oscillates a little at the last step, and then it does get the classifier and it stops. So this is the perceptron. It does work. I mean, and versions of it are used, or at least were used very sort of reasonably heavily till a few years ago, until deep learning took over. So yeah, but still, you will see that it is part of deep learning story also. Okay, so this is the perceptron. The problem with that is one perceptron you cannot do much, and this has been mathematically proven. It's not hard to prove mathematically. You cannot. There are certain very simple functions you cannot solve. Are there an odd number of ones? So there are things like this that you cannot do. So what you want to do is you want to connect them, and this is also an idea that is coming in some sense from biology. That I mean, the human brain is connected. It does not is not one neuron or any other brain. So you want to connect them into a network, and maybe then it can do things. Okay, so here is so now we come to the price. So the question is to learn the network of perceptrons, and there are these two papers which actually have very similar results. One is by Shuichi Amari from 1972. The other one is by John Hopfield from 1982. The Hopfield paper, I think both are mentioned in the So okay, good. So yeah, so here is what Hoffield and Amari, 10 years apart, were trying to do. So So the Ising model can be seen, now that I've shown you the formula for the perceptron, is a network of perceptrons. Each vertex is actually a perceptron. And there are the energies. So here is how they were trying to model memory. So before that, I should say one thing, so this is also called the spin glass, and that also is like around the same time. So the Sherrington-Kirkpatrick paper also, I think, is between like maybe 6 years after Amari, 4 years before. Okay, except for that aside. So here is what they want to do. So given some pattern sigma, so some assignment of plus and minus to the nodes. So this is the pattern. This is the pattern they want their network to remember. So what does it mean to remember? So to remember means that if someone later on comes to you, and this actually Hoffield has a very nice example, says that here's a paper. His paper starts like this. Here's a paper which is written by someone and Vanier. And actually it was written by Wanier, but still you can probably guess that, oh, this is the one if you have seen the paper. So this is what it means to remember, right? Like even if you get a slightly noisy version of this, you can figure out what was there. So this is what they want to do. This is what they want the network to do. So what will the network learn? It will find some J. And then maybe later on, someone will come with a different pattern, which is different, but not too different. And then the network should be able to tell that this is what I remember. So in this way, so the J will be there. This J will be such that it will minimize the total energy. So that seems intuitive, that if you want to learn sigma, you should sort of get the network to a state which is the minimum energy for that sigma. So that sigma would be minimum energy with that J. So that's the J you will choose. And now what you want to do is the following. Suppose that this J is fixed. Now we don't remember, or at least I don't remember sigma. The network does in some way. Someone else comes with this new pattern tau, which is slightly different from sigma, but so not very different, but slightly different. What they want to do is to get the network to sigma. And how does the network do that? It will just do very local computation. Each vertex will just look at its neighbors and try to minimize its local contribution to the energy. They will keep doing it again and again, completely mindlessly, and under reasonable conditions. When things are close, they show that this will converge. So Amari proved that it will converge mathematically in his model. Hoffield did simulations. Their model is slightly different. Amari's model is that all the vertices do simultaneous updates. Hoffield's model is that so it's essentially like at each time one vertex is chosen at random and that vertex updates. So these things also, but they should be morally the same. So this is what they did. Okay, so then there were several later extensions. This is all about binary things. Hoffield and Tank did many extensions to especially the analog setting. So now it's not just plus and minus, but everything has to be continuous, and still you want to get this kind of discrete behavior out of it. So they're connected to the theory of dynamical systems. This is what this was. Okay, but there's a problem. So at least we are learning. So we're learning with some kinds of networks, but there is some problem. So we are usually not interested in storing one pattern, right? We are interested in storing a class of patterns. So what do I mean? So if I tell you that I have a model which will detect the 1, 0 I drew yesterday and nothing else, you're probably not going to be very impressed with such a model, right? I mean, why are you interested in the 0 I drew? Right? But if I tell you that I have a model that will detect zeros that any person who writes in, say, English or Marathi or Hindi wrote, and it will detect that, oh, this is a 0 or not, then maybe you're somewhat more interested. Maybe then you can use it to detect pin codes. This is actually done. This is one of the first examples people try to sort of completely solve using these methods, not just 0, all the digits. And by now you can do that. Like you can from scratch write that code. Over a few hours. So maybe after the talk, that's an exercise to do that. But okay, so this, of course, the previous one does not do, because the class is huge. I mean, all the zeros look similar, but they're not the same. They're not just small changes, like a very few pixels different from each other. Right? That's not going to be. They look as shapes very similar, but not in terms of this pixels thing. And that's what the previous thing was doing. So this could potentially be solved by not just looking at the lowest energy configuration. So this is what we are doing. So you also want to look at all the nearby states. And this, again, if you have seen any kind of statistical mechanics, is clear what you want to do first. You want to do a Gibbs distribution. You want to not look at the so-called zero temperature model. So the zero temperature model will be in the lowest energy state. You want it to have some sort of, it should be a heated up model. So it can go elsewhere also. You can slightly perturb it around. So that's so that would be one thing. So you will now have a probability distribution over situations, which will be somewhat biased towards the low energy states, depending upon what the temperature is. But it will not just be supported on the lowest state. So that's good. So this is one. But even these might be too regular. Like, I mean, you can imagine that this kind of distributions have very strict properties. They can only do certain things. And this is true in the easing case. So this may not be like, this might be too regular to capture the kind of rough things you want to capture. People writing zeros and ones and twos, things like that. The images will not have all those regularity properties. So here is this one of the other things. So this is Fallman, Hinton, and Sinofsky in 83. They proposed something that they call Boltzmann machines. This nicely combines various ideas. So it's a method to solve both these problems that I was writing down in one go. So use the positive temperature version. This is regularity issue. So what do? So this is sort of a certainly interesting point. It's sort of, again, one of those things which is obvious in hindsight. It's like you say that the network, so far our network was just everything was being visible. So all the inputs we were seeing, all the outputs were all visible. Now we let the network have some computation space. So there are some nodes the network has which are not part of the input, not part of the input. So neither the input nor the output. They are just there for the network to do its own computation. Okay, so this is also true for the brain, right? I mean, it's not like you just have an input-output circuit. Something in the middle, you have all your memories, all things you've learned and so on. So this seems like a very natural thing to do. This is also true for any kind of reasonable model of computation. You have seen Turing machines or anything, or even the computer, right? It has things that are not just input and output. There are other things where it can do some calculations. So this was important. But how do you do it in this restricted easing model setting? So they said, oh, very natural. What you do is you just make these hidden nodes. On the full space, it is the easing-like distribution. You learn all of those things there, but you will only at the end look at the so-called marginal on the observed nodes. So it will be some so-called what is called a, so it's like I have this easing-like distribution on the 100 nodes, but I'm only going to look at 20 of those nodes for my output. So the distribution on these 20 nodes is not going to look anything like an easing model. So now those restrictions are in some sense gone, and potentially you can model many more things. So this power, of course, comes at a cost. Now it's harder to find this J. I didn't tell you how to find the J in the Amari et al. Setting also, Amari and Hopfield setting also. But there it's sort of believe that it's easy. Here it's somewhat harder. So they did come up with an algorithm, but it is, so Boltzmann machines are not used today. And one reason is that they are computationally not as nice as the earlier perceptron things. So things that are used today are more like perceptrons connected to each other rather than Boltzmann machines connected to each other. Okay, so here is a picture from the Swedish Academy of Sciences. It's free for non-commercial use, so I thought I'll use it. It's not commercial. So it's a good summary of the three kinds of networks. So there was the Hopfield network or the Amari-Hopfield network. Everything was very discrete. Then there was the Boltzmann machine, which is this easing model type thing with randomness and some part possibly hidden. And then there's something also called the restricted Boltzmann machine. This was intermediate thing, also very important, also mentioned in the citation. It's not used so much today because other things are found to be more efficient, but the ideas that went into it did influence what is done today. Okay, so this is sort of like you restrict what kind of connections are allowed in the Boltzmann machine to make it computationally more feasible. Okay, so what we want to do now is that we want to get into the, like, just jump directly into the deep end. So everything so far was very, like, the only thing I showed where something was deep was this, there were some layers. But when you hear neural networks, you hear words like deep learning, very deep learning, I don't know what. And so usually, like, there are many layers. So you have, so it's like a perceptron, then maybe a perceptron taking input from other perceptron, then that layer also giving it to another perceptron and so on. So depth just means this. So you're taking something simple and then composing it several times. So with hidden layers like this, the input and output, so this kind of thing had a long history. Boltzmann machines are not the first one. There are several things before that also. But so you can look at this survey. But the issue is, again, how do you learn all these weights? There will be weights at all these layers now. All these different layers are doing their perceptron type thing, and they are all interacting with each other. How do you do these weights? So the first method is to try to do what you probably again saw in class 10 or 12. This is like while doing linear regression, which is to optimize using gradient descent. Maybe there it was not phrased like that, but that's what it is. But so you try to compute the derivative of some kind of a loss function, like how far are from the output, compute the derivative and do it. Very basic class 11 or class 12 type stuff. The question is, how do you compute the derivative, right? This is a big network. So let's see. The obvious thing to do is to you say, okay, I want to compute the derivative. This has been known at least in Leibniz. It's called the chain rule. So here is you try to apply the chain rule and you go ahead and apply it. It's called forward mode. So you say successively compute from input to output a derivative of the output of each layer with respect to the weights. And you quickly see that this runs into a problem. Like try to compute. So the last one is of course of your, but the intermediate results can be very large. The point is that just look at this. What is the output of the third layer? So this one here, with respect to say W1. Actually, no, it should be W2 probably. So it will be 50 cross 400 into 50 because there are 50 output dimensions, and there is you're looking at the derivative of 50 dimensional function with respect to 400 to 50 variables. So all of those things will come into the. So you have to combine all of this. In the end, you'll get a small thing, but all of these things will be there. Okay, so it's too expensive. So if there's a forward mode, there should be a reverse mode, and that's what you do. There's something called reverse mode, and what is this? You successively compute the derivatives. Now in the opposite way, you look at the derivative of L with respect to the W4 first. So instead of coming from like top down, you go from bottom up. So the derivative of the loss with respect to this, then with respect to this, then with respect to this. And this time, the crucial thing is that the loss is just a number. You're only computing these vectors. So all of them will only be of the size proportional to the variable with respect to which you're computing the derivative. So essentially, all the intermediate results will be the same order of size as the network itself. This is extremely important because the networks, especially with these big networks. Okay, so let me just conclude. So this is reverse mode, and so reverse mode is important. It is you can see this something where people call it the cheap gradient principle. Gradients are cheap, but there's also something called the cheap Jacobian principle, which is false. So you cannot compute Jacobians quickly, and that was why the forward mode was sort of failing. It is so important that it has been discovered many times from the 50s onwards. So it has been discovered in chemical engineering, circuit design, numerical analysis. In fact, this is a very interesting story of this Lin 76 paper. It's actually a thesis from 1970. He was a computer scientist who was for some reason doing numerical analysis who sort of did this like this. There is for my colleagues, they might be more familiar with Bauer-Strassen. It's a fundamental result in algebraic complexity, which is based on this. But there are many similar things. There's also this line. There's also a close connection to backpropagation in neural networks, and it's cited to a paper in '88. So this comes from a paper which actually gives the name that is, maybe you have heard, reverse mode AD by backpropagation. Okay, so it's called backpropagation in the neural network literature, and this is the paper in Nature that made it popular. It's from '86, quite after all of these things, but it was one of the first to apply it to neural networks and also do one more thing. It did one more very interesting thing, which is it actually tried to interpret what these layers are learning. Maybe there's a final output, but what are the intermediate things that are being learned? So that's it. So training of large models essentially will be impossible without this principle. It's a very simple principle. It's available today. So they say that it's available to everyone in the field of scientific computing in 2008. Now it's even more available because there are things like TensorFlow, JAX, PyTorch will do it automatically for you. To try it out, quite nice. And so let me just end with pictures of two deep networks which are very popular. One is the so-called ResNet. It's an image recognition network. The one on this is what underlies many of these large language models. You can see that they are deep. So you need to compute derivatives, and for more application. So we'll see more applications of some of these more deep things and the gradient computation in the next talk. So stop with that.
Thank you very much. Piyush, questions? Okay, that's fine. Are there any questions? Yes. Ravi,
Hi, Piyush. Very nice talk. That's a quick question on the optimality.
Yeah.
So when you construct, suppose layers, and you are showing this IC model or so on,
yeah,
what is the optimality regime or number of layers which can be constructed? How will I choose an optimality in this problem?
Yeah, so you do something called it has a fancy name, hyperparameter optimization, which basically means you try out several things and see which one works.
But on if I come from a physics background, I start writing Ising model and I write my degrees of freedom and degrees of symmetries.
Yeah, so easing model you can use the easing model, yes, but like for general, like if, I mean the usual kind of problem they will be doing with say some image recognition. So there you will try out 3, 4, 5. I mean, so there will be some intuition that people get from just having tried many things before, but there is very little theory to guide.
Like there is no physics principle I can apply to.
Yeah, so there are some that people try to like, so at some levels like they say like how should you initialize things, what should be the relative order of the weights. So there are results like that using essentially random matrix theory kind of ideas. But yeah, so okay, so I didn't say any outstanding problems. Everything is outstanding. There is very little theoretical explanation for most of it. So almost everything is outstanding.
Hi, right here.
Yeah.
Just very basic. What was the innovative step or sort of aha discovery, or what was the sort of
Yes, so I think I showed a picture in the beginning. It's not Michelangelo, it's Kailash temple, right? So it was brick by brick building. With you remove one and the whole thing would not exist. So it's more like over about 70 years.
They finally achieved success.
Yeah, several things happened, and like I mean, there are also a lot of engineering things that happened, like faster processors, more energy efficient processors, which also fed into this.
But no sort of single thing that enabled this now.
Yeah. So there is no, I mean, so there's no now as such. I mean, like, so I mean, I don't know why. So I don't know what the people in Stockholm were thinking, but yeah. So the idea is that that I mean, the deep learning thing, the deep learning achievements have been coming over the last 12 years, and yeah. So.
There's one more question.
Yeah. So you mentioned that the two important things the system should have is one, it should consume data like in a small chunks, and it should move slowly. Like,
I shouldn't say should, but this is what the rule of thumb is that people try to follow.
Yeah. Why is it important?
Yeah. So okay. So the thing is like, so it has to do with the thing that you don't have a good theory for what the model is doing. So I mean, okay, maybe some of my colleagues can help with this, but the point is that you don't want to commit at any one point to optimize for one input, because you don't have a physical model that this is what is happening. So you want to be sort of circumspect. If some other example, it's not like to just solve this example, I make a lot of errors on the other examples. So again, yeah, so these kind of ideas have also been there. It's like it's the basic, so the thing to search for is something called stochastic gradient descent, which has been used in other systems.
So there's been criticism why this is a physics prize. What's your take?
Oh, why
is this a physics Nobel Prize? There's been a lot of criticism in the press.
I mean, like
what physics do you?
So I remember that a joke that was told to me when I was doing a PhD. There was in our university the physics qualifier exam. The syllabus was written as 2 lines: classical physics and modern physics. So if you want, yeah, it's physics. Right? I mean, it's about the world. So.
No, I agree, but I mean, not. Well, that's what I hope, but without trivializing it.
Yeah, no.
What is the hard physics principle that stems to the question that's
also. Yeah, so okay, so hard physics principle, I don't think there is. So it's not like there is any one thing you can point to that this was a. But many of these ideas, like as you can see, these people were inspired partly from the statistical mechanics ideas of energy.
Rather remote, yeah.
Yeah. So yeah, you can.
Let's thank Piyush and move on to the next prize.
Sorry. Yeah. So just so there is something called the MD data set of digits 0, 1 to 10. Try to write a 3-layer multi-layer perceptron with say 3 layers to detect those digits. You just search for MNIST database. It's a standard database.
Okay, so the final presentation is going to be by Venkatramani from DCS, and is going to talk about the Nobel Prize in Chemistry from computational protein design and for protein structure prediction. Ravi.
Hello, can you hear me? Can the people at the back hear me? Yeah, great. Thanks, Jyotishman. I have no confidence I'll finish this in half an hour actually, but please warn me. Right. Okay, so the 2024 Nobel Prize in Chemistry was actually given to in 2 parts, right? So the first part was given to Professor David Baker from the University of Washington for computational protein design. And the second part, which was shared in equal proportions between Sir Dennis Hassabis and Dr. John Jumper from Google DeepMind, was for protein structure prediction actually. So the reason the prize was given to these guys is because they opened up
a window, or actually, in fact, they opened up the protein universe for us. Overall, they described the structure of every known protein to human beings. And in addition, they also kind of enabled us to design proteins which are completely unknown to nature, right? So this is really a fantastic, breakthrough. So I'll just take you through all the essence of it, actually. So the first, I'll start with a very basic question, basically. What are proteins? Because it's a very diverse audience. And why do we care about them? So proteins are large biological molecules, right? And essentially, they carry out all the essential functions for life. Inside our body right now, there are about 20,000 different proteins working to keep us alive. Here are some mugshots of some of my favorite proteins. This particular protein is an enzyme. Essentially, this actually is a detox enzyme. It creates, it takes harmful chemicals that enter our body and flushes it out by making it water soluble. This is hemoglobin, which all of you will recognize is, a famous transporter. This protein on top is rhodopsin. Essentially, it is the molecule which is responsible for vision, how we see. So essentially, I cannot emphasize how crucial it is to visualize these proteins in this way. What I've drawn here in these structures. These structures are the starting point for understanding how these proteins function. And essentially, for all fundamental, questions in chemistry and biology. Additionally, if you think about it from the applications perspective, they're absolutely critical if you want to design drugs which actually, target these proteins. In fact, nearly all medicinal drugs basically target proteins. So it's absolutely important from a practical viewpoint. Now, I just want you to gain a deeper appreciation for what you're seeing here. These images, these are all protein structures which are atomistic detail, right? But what are we seeing here exactly? So I just want to take you through a few basic slides and terminology so that you're clear about the importance of what was discovered. So right at the beginning, from what Ullas mentioned, basically everything starts at DNA. DNA is the blueprint for life. And we understand essentially what DNA, essentially, it carries a genetic code, right? But what does it code for? So the code, basically instruction set is packaged in sets of 3 base pairs, and it's called a codon. Each codon actually pretty much has instructions to make a simple amino acid molecule. So the amino acid molecule pretty much has this standard structure. Every amino acid actually has these nitrogen atoms, carbon atoms, oxygen atoms, and hydrogen atoms. But in addition, there is also something called a functional group, R group, which is different. So different amino acids actually differ in terms of their R group, and there are 20 possible such R groups. These amino acids actually all, they are like molecular, they're basically building blocks. They come together like Lego, and these particular backbones can actually bond together, and you can actually form dipeptides. And if you actually have more of them bonding together, you actually form something called a polypeptide chain. Now, you see the different colors. So there are 20 possible colors, as I mentioned, depending on the R group, right? And the sequence in which these colors are actually, the order in which these amino acids are present in a chain, that is called the protein sequence, right? So this is going to be a central player. The protein sequence is one of the primary pieces of information that you can have about a protein. Because the Human Genome Project and the Genome Project was way ahead of the Proteome Project, essentially pretty much all the genetic information out there. And from this genetic information, you can very easily obtain the sequence information rapidly. In fact, that's already been done overall. Now we go to structure. If you take this particular sequence, that polypeptide chain, and just drop it into solution or something like that, then these chains can actually adopt different kinds of 3-dimensional shapes. Okay. So now here I've shown you one particular shape. So here I have hidden the R groups, and I'm showing you only the backbone. And these backbones are arranging in this planar configuration, really, and there are some interactions between these chains which keep them rigid. So these are what we call beta sheets. On the computer, you can actually draw them in terms of this cartoon, which is a little bit more human-friendly for us to identify them. Sometimes the chains actually adopt this helical conformation, and you actually can draw them on the computer as well. So if I have a protein like this is also one of the proteins that I worked with. It's a DNA polymerase. And if I show it in all its glory with all the atoms, it will look like this. But if I draw it on the computer in this cartoon form, basically it looks like this. Helices and sheets and so on and so forth. You can see nuances here in this case. How are these structures determined? Right? Whatever I showed, these mugshots. So the gold standard is X-ray crystallography. Why is it a gold standard? Because of resolution. This is a technique that has the best resolution for viewing the protein structures. So basically, you grow protein crystals, you bombard them with X-rays, you get this diffraction pattern. From the diffraction pattern, you get electron density maps. and then you fit this electron density map using molecular modeling and with multiple terms of refinement, basically you get this protein structure. So that's how those structures were obtained. You can also do it using NMR. You actually have a protein solution, you expose it to magnetic fields, expose it to radio waves, and then you activate the spin degrees of freedom here essentially, and here you're looking at essentially the resonant frequencies, spin degrees of freedom, plotted in 2 dimensions. The diagonal terms here show the atom types. Off-diagonal terms give you distance constraints between the atoms. So using these constraints, again, you can do molecular modeling, right? And from the molecular models, we'll get an atomistic level model of this protein structure. Again, what I showed you earlier. Wonderful. So we have a technique to do this basically. So what's the problem? Look at here, the cost. For a single protein, it can take months to years to get this kind of a structure. Equipment cost is about 1 crore or higher at this point. And I have not mentioned one more technique called cryo-EM basically, which is even more expensive, but also can give you fantastic structures basically, but the resolution doesn't approach these 2 techniques yet. Using these 2 techniques over the last half a century of work, I was talking to Sandhya Kaushika, basically, it's basically elbow grease, right? You have about 250,000 structures, 0.2 million structures that have been solved and deposited in a protein data bank, right? This forms a repository. Until a few years ago, this was a repository of protein structures. The problem? I told you about sequence of protein, right? The sequence information, the number of protein sequences for which we have information is about 200 million. The number of structures that are solved is 0.2 million in this case. So I would say a few years ago, basically, if you were saying, oh, neither me nor my grandchildren will ever witness all the proteins that are lying, basically, you would not be very far off using this kind of a pace. This is what our Nobel laureates have actually solved, right? And they've opened the protein universe for us. I'll tell you how. Okay, so first, the question that I want to ask you here at this point is, so this was already very, very slow. So the question is, since we know 200, 300 years of chemistry and physics, why don't you put it into the computer, and we also have biology, right? And why don't we from the computer try to figure it out, the overall thing basically. But people have tried using that essentially, but it didn't work, right? So there was something missing, right? But that something missing was actually already discovered a long time back, just that people didn't, put it together. And this is, it goes through a pioneering experiment in the 1970s, basically. So in 1960s, in fact. So C.B. Anfinsen, who got the 1972 Nobel Prize for this remarkable experiment, showed that if you take a protein, right, and subject it to conditions where the protein opens up into its polypeptide chain, its primary sequence, right, then it loses its function. But then if you reverse those conditions, right, here I've shown temperature as the condition, basically. You heat the protein and it goes into this chain. If you then cool the protein down, then this chain comes back to its original structure, that exact same structure that it went from overall, right? This is amazing. So what he came up with a hypothesis, basically, it's called the Anfinsen's dogma. Essentially, the sequence of a polypeptide chain contains all the information for it to fold into this final state. Now, you should merge this information with something called Levinthal's paradox. Okay, so think about it, basically. Suppose you have a protein with 100 amino acids, and let's say each amino acid has 3 degrees of freedom, 3 states that it can sample, for example. Even then, you have something like 10 raised to 47 possible states of the protein. So if you assume that the protein was actually randomly sampling these states, right, and atoms move at a time scale of picoseconds, for example. So let's say 10 raised to 13 states were sampled per second. Even then, it will take you longer than the age of the universe to actually find this particular folded form, right? So the point of the matter is, basically, if you put these 2 together, the sequence contains information about the folding, and this search is not a random search. It's a directed search. There is an underlying energy landscape, right, which directs essentially the protein to go from the unfolded state to the folded states. This hypothesis is, this dogma is also called the thermodynamic hypothesis for this particular reason in this case. So the sequence contains that information, and it also knows there is some information about the underlying energy landscape in terms of potential or statistical potential or whatever it is, which tells us how to go to the folded state. So this is the key. So if we understand like the basic driving forces, the question is, can we predict the 3D structure from the sequence alone, right? Without going through all the in-between steps of protein folding. So this is what our laureates have done, right? Demis and John have actually pretty much harnessed Anfinsen's dogma. So they've shown us essentially how you can take a sequence and from that directly predict accurately this particular 3D structure of the protein. David Baker, on the other hand, has flipped Anfinson's dogma. He has very different question. He's asking, suppose I know what structure I want. I design the structure on paper, for example. Can you find me a sequence which is going to fold into that state? Right? So this is protein design overall. So these are the two parts for which the Nobel Prize is given. So I will first take David Baker's work because chronologically, surprisingly, you would imagine that you would solve the top problem first and then come for the bottom problem. But surprisingly, the bottom problem got solved first. So I'll go chronologically in that order, actually. So what David's strategy was the following, basically. He decided that he was going to understand essentially all the basic rules, the chemistry and the physics, the chemical physics of the particular problem, right? So essentially, if you take a sequence, I showed you all these amino acids, right? So these are all labels for different types of amino acids that are present here, in this case. And if you look at the chemical properties, all these amino acids have very unique and interesting chemical properties, actually. Not unique, I'm sorry. They have different types of chemical properties. Some of the amino acids are hydrophobic. They hate water, right? They run away from water. Some of the amino acids are hydrophilic. They love water. They like to be solvated. They like to bond with other amino acids, for example. Some have positive charge on them. Some have negative charge on them. There are sulfur-containing groups, right? So with Arnab, we did a chai and why basically where we played this game, you cook up a random sequence of protein, right? And then you essentially color them according to these classifications. And then you start with an unfolded chain and then you ask, oh, how is this thing going to fold, right? So then if there are some rules, then you can actually fold the protein into some particular form in that case. So I'm just making some example rules up, basically, but the rules are far more complicated than this. So for example, if my rule was, okay, sulfur bonds have the highest weightage, for example. So here I look at things that have sulfur bonds. Cysteine and cysteine, they have sulfur. So I know that in this change, this cysteine and this cysteine have to be close by for them to bond and form a sulfur bond. That gives me a distance constraint. Ionic bonds. So if there's a positive charge and a negative charge, they can actually form something called a salt bridge, electrostatics. So in this case, there is a lysine here, and then there's an aspartic and glutamic acid here, basically. So these have to form like some kind of a salt bridge. Again, that gives you some kind of a constraint. Hydrophobic collapse. This is a more complicated thing. All these groups, all these black groups actually are like, they hate water. So they would all like to be buried inside the interior of the protein, right? So these rules tell you essentially which amino acids are inside the protein, which amino acids are outside, which amino acids are talking to each other. These are all constraints on the degrees of freedom that a protein can adopt in that case. So that's what we mean that the primary sequence actually contains this information. So this is what David Baker did. I mean, so starting from 1999 to 2003, he wrote a series of papers basically where he came up with a general protein design algorithm basically. So this algorithm, of course, has grown sophisticated with time basically, but the essential principles remain the same. So the idea is essentially you drop a protein structure on paper, what kind of a protein structure you want, right? And this protein structure may have those alpha helices, they may have those sheets, and you want them to be placed right next to each other. That's it. You draw it on paper. Once you draw it on paper, then you know that you have certain number of degrees of freedom, right? And there are constraints based on those rules that I just mentioned to you actually in that case, right? So once you have those rules and constraints, then you actually make a scan of the protein data bank, etc., asking how many sequences actually follow those constraints of these kind of structures that can be obtained. So you can come up with an initial sequence, a guess sequence, and this guess sequence actually is supposed to give me a 3D structure. So these guess sequence with the degrees of freedom and constraints can be used to cook up a 3D structure. So once you have the 3D structure, you want to rank it and score it. How good is this 3D structure? So you need an energy function to score it. So this is a crucial step. You need a proper energy function actually, and nobody knows what the underlying potential energy is. Nobody knows the exact term. So this is the energy function that he came up with actually. It's horrendous. If I were to look at the physicists in the audience, this would be considered very inelegant. I would say basically there are 11 terms here, right? But the important part is that among these 11 terms, there are many terms which are based on physics. Like for example, there is Leonard-Jones interactions. There is electrostatics, of course. There is solvation, right? In addition, there are also empirical things. For example, there are energies associated with torsion angles, for example. And these come from the principles of biophysics, 10 minutes. Okay. These come from the principles of biophysics, where essentially you do simulations and you find out essentially what kind of probabilities various kind of torsion angles adopt. In addition, you can also go to the protein data bank and find out what is the distribution of torsion angles that are existing in protein structures now, and come up with a probability distribution function and then invert it to get energies in that case. So you can compose an energy functional like this, and this energy function then can be used to score this 3D structure. And then if the score is whatever it is, you try to improve upon the score by repeated refinements. All these steps are actually coupled by Monte Carlo optimization in this case. Now, the only key point I want to make is all these terms here, including the weights, etc., they are actually learned from experimental data as well as simulations, and the potential is chemically detailed. Like even a single carbon atom can be in different forms based on which chain it is, or like whether it is an aliphatic or aromatic actually. So it's incredibly detailed actually. So what are the first? Essentially, this algorithm was the first one to design a new protein completely from scratch, a de novo protein design algorithm. It is applicable for proteins of any size. So this particular initial protein that they designed was about 93 amino acids, which is considered large at that time, but now it can be extended to very, very big systems as well in that case. In addition to sampling the side chains, this was the first algorithm which also sampled the backbone degrees of freedom, basically, right? And ultimately, this was the first algorithm which had a self-consistent sequence as well as structure optimization. You want to walk in sequence space, and also you want to walk in the structure space, and then self-consistently optimize both to get the result in that case. The result of that was this first protein in 2003, which is called TOP7, right? It has these things are helices, these two cylinders, and these are the beta sheets for this particular protein in this case. So this is a first large protein with mixed alpha beta topology. The structure was validated with X-ray techniques. This fold is unlike any structure that exists in the protein data bank, right? Which was solved. And the sequence is unlike anything that we know from our sequence analysis, etc. So that's one reason basically why this is such a major landmark. Since then, I mean, he has designed enzymes, ligand binders, essentially the proteins which actually bind to steroids, etc., with incredibly high affinity and selectivity. And also self-assemblers. You make these protein fragments and drop them in solution, and they self-assemble into these beautiful structures, for example. In addition, sensors, switches, and there are more things coming at this point, basically, overall. So incredible piece of work, and for which I think he totally deserved the Nobel in this case. Now I move to the second part, right? The Anfinson's dogma parts, from the sequence to the structure. How much time do I have? I think it will be okay. Okay, so for this, I mean, I want to start the story not with directly the method, but essentially how AlphaFold actually came into prominence. It's a very fascinating story. So there is a competition called CASP, right? It's a biannual competition where groups all over the world compete to predict the structure of a protein from sequence, right? It's a very high profile contest. And for example, CASP 14, which was held in 2020, had about 97 groups from the world, 19 countries, 215 methods, and they were targeting 100 experimental targets. These experimental targets are also unique. The organizers contact experimentalists. They tell them that basically we want structures. So these guys solve the structures, but they do not publish them. The structures are withheld until the competition actually finishes, and then the structures are revealed in that particular case. So now here, this plot is very detailed, but I'll lead you through it basically. So essentially on the x-axis here, we have different types of protein structures, easy to difficult. Right? Easy means the target structure actually already has known analogs in the protein data bank. There is already structural information available. So essentially all you have to do is mix and match. You see the sequence and you see the structures in the protein data bank and you mix and match basically. Right? Difficult is where there is no precedence, very little information available for the structure in that case. On the y-axis, you have a score. 0 means, I mean, like, your prediction is ridiculous, is not matching at all, and 100 means, I mean, you have perfect 100% match. In this particular case, 90% is the threshold where everybody wants to target. Why 90% and why not 100%? Because experiments also have errors. And so 100% doesn't mean anything in this particular context. So now, look at this data here. So what this is showing is essentially how the CASP groups have fared over the years since the original CASP from 1994. So you can see almost from the original CASP, right? The easy targets have already been at 90%. People can get that very easily. But the difficult targets have been stuck here up to 40%, below 40%, right? Right here, there's an exception basically. Something has happened around here. So what are these lines? These lines belong to CASP 13, the previous CASP from CASP 14. And the difference between this and this set is that here there are AI-based methods that have started being applied. In fact, AlphaFold actually has entered the competition here, and AlphaFold has actually won this competition, being at this level in this case. And this is what Sir Dennis Hassabis, I'm sorry, his contribution was. Essentially, he put AlphaFold on the map for winning this CASP using AI methods. This was the first demonstration that AI could do something like this, which is and much more accurately than everybody else. And this jump from here to these white dots is coming because of the inclusion of Dr. John Jumper. Right? So when John Jumper joined Google DeepMind, he brought with him his expertise in simulations and protein biophysics, as well as biochemistry. And since he had that, they improved the algorithm significantly using that knowledge base, basically, and they reached close to 90% for even the difficult targets in this particular case. Right? So here is a particular one target which is considered extremely difficult. And essentially, you can see the structural overlap between the experimental and the DeepMind, and essentially it is really matching. Here is something even more amazing, in my opinion. Here, they've compared the score of Google DeepMind, of AlphaFold's submissions with respect to structures with decreasing order of resolutions, 1 to 2 angstroms, 2 to 3 angstroms, greater than 3 angstroms, cryo-EM, etc. And you can see that the score is going down. That means the score is actually sensitive to the experimental resolution. So this only happens if what you consider as the gold standard is no longer the gold standard. The structures are more accurate than the experimental structures, right? And then the resolution is going down. I'm not going to read all this out for you, basically, but please read this last point, basically. So even experimentalists who were contacted sometimes could not solve the structures. So AlphaFold actually predicted the structures and they sent it to them, and then they were able to solve the structures. So we are shocked, stunned by the quality of the model. You would not believe how much effort we have put into getting the structures. Years of work, both cryo-EM and crystallography. I mean, this is really shocking. Right? But the kicker is still yet to come, right? So how does the whole thing process work? Do I have time for this?
Yeah.
Does everybody want to hear this? How it works? Yeah, okay. All right. So essentially, you start with an input sequence, right? Let's say you want a human protein structure, and there's the human protein input sequence. The program starts doing two kinds of searches. One is it searches the genetic database for this particular protein sequence in all organisms, not just humans, but all organisms. Hen, fish, whatever you can consider. And then it does something called multiple sequence alignment. See, it's a particular protein. It is in all these different organisms, performs the same function. So its structure and its features must be similar because it does the same job overall, right? There are differences. But if you align these two sequences, you should be able to see, the matches of this. So this is called multiple sequence alignment, actually. And what does multiple sequence alignment provide? So the point is this, basically, that consider the sequence in my body versus that in a hen, for example. It's the same protein performing the same function, but the amino acid chains, right, they're not exactly identical. Suppose I have a positively charged residue at a particular point, and it is making a salt bridge with a negatively charged residue. In the hen, maybe that positively charged residue gets changed to a hydrophobic residue, which is running away from water. If this happens, then the negatively charged residue, which is out there, should also change. This is called co-evolution. If one residue changes, another residue also has to change, basically. And then if these two residues change simultaneously, it means they are close to each other. They are proximal. So by doing this multiple sequence alignment, you actually get distance constraints. In addition, it also does a scan of structure database search and actually finds out similar protein sequences and finds protein structures which are similar looking. And from that, it can actually create a distance map here in this particular case. You take this multiple sequence alignment and distance map and you pass it to this block called evoformer. I mean, in short, what the evoformer does is basically it actually takes the multiple sequence information and the distance constraint and exchanges information between them and optimizes them periodically using those weights. It learns how to guess what the correct distance matrix is, basically. And once you get that, then you're able to generate a structure model. Again, this is a very simplified explanation, but there are a lot of fantastic algorithmic advances here. And from this, you actually create a structure which comes out, and then with a few more refinement models, basically you get the final structure. This is the kicker. For a single protein, 15 minutes to 0.5 days. Compare it with the months to years I talked about with cryo-EM and, with the other techniques in that case. Equipment cost is 10 lakhs. I can do it on my desktop computer, right under my desk in this case. So the big deal is that before AlphaFold, within 2 years of Cas14, this is the structure, this is the space that we were with experiments, 0.2 million structures. Now we have all the 200 million structures known to man, known to human beings, all of them. Right? And this AlphaFold, previously 1 million structures, is when it won that Cas 13 contest or whatever it is in that case. Not only that, you now have structures for over 1 million species. From animals, plants, bacteria, fungi, etc. These small circles are what we had before. Right? And this is AlphaFold right now, actually. So I hope you'll agree with me that our laureates have opened up a completely new world to us. What we do with all this information is completely up to our creative imagining at this point. Right? It's truly novel worthy. Thank you.
Very nice, Ravi. Thank you for that wonderful summary. Questions for Ravi? Yeah.
Can you have two different structures from the same configuration of acids or proteins?
So what you're asking me is the same sequence.
Same sequence will get you two different like 3D structure. Can you have that solution?
Yeah. So there's an excellent point actually. Protein. So what I'm showing you are snapshots of the protein. It's like showing you photographs of a human being, right? So you know that that's not going to be true because human beings move. Proteins also move. They do thermal motions, for example. So the underlying landscape of proteins has a global minimum. And it has many other minimas as well, right? So sometimes those minimas are actually even useful to function. What this is giving you is a global minimum, right? But there might be other structures that the protein can also access with the same sequence, 3D structures. So that's a.
So at a time, it is giving you the global minima configuration, not any minima.
Yeah.
Okay.
Right, yeah. And actually, the question is a little bit more. The answer is a little more subtle than that actually. So you see, this is a learning algorithm. Right? So learning is based on some training set that is provided to you. So the training set has like various things into it, hidden into it, like, the biological context. Let's suppose that I was actually getting the structures from only X-ray crystallography, and the algorithm was learning structures from X-ray crystallography. The new structures that it will predict will also be like X-ray crystallography then in that case, right? But if I had like some other technique, for example, and I got like structures which are slightly different. Like NMR is a solution state technique, basically. Sometimes the structures of NMR differ from that from X-ray crystallography in that case. Then the predicted structures will also have that imprint. So these are some nuances that one has to keep in mind. Yeah.
So you are saying that experimentally determined structure is about 0.2 million. And we now, with this intelligent algorithm, we know effectively 200 million structures.
That's right.
So is it still important to keep on determining structures experimentally, or we have determined enough, now we can just go ahead with this?
I would say it is still important to carry out experiments. The reason is that so there was a useful review. There was a very nice review article that just came out recently, actually, which actually talked about the relevance of AlphaFold database, actually. So the way you should think about the AlphaFold database is it's one of the most powerful hypotheses we have about protein structures, right? So if you actually use experiments and determine the thing, basically then the hypothesis gets validated. So earlier we had nothing. Now we actually have a hypothesis. And if you do experimentally determine the structures, then you're able to determine them under different conditions.
So it should be seen more as a guide to experiments rather than a definitive structure.
It'll be, right, absolutely.
Yeah, actually, my question was very similar, but so, I mean, learning depends on having an extensive database of experimental structures, right? I mean, all of this was possible because the database was
So
as huge as it was.
Yeah, I'll just kind of, let me just go back to this slide here, and it'll actually tell you something which is nice and interesting. Okay, so look at this final structure, right? This final structure has different colors. right? So what this is telling you is AlphaFold also tells you how confident it is on a particular fold of the protein. So there are some folds parts of the protein which has very high confidence. Some parts have very low confidence, for example, in that case, right? So now, depending on essentially the confidence level, you can also estimate that if you are actually going to solve something with X-ray crystallography, right? How close it is going to be, that's going to be to this AlphaFold structure. For example, if AlphaFold has predicted something, but the confidence is very low in general, it makes sense for you to target that experimentally, right? Those are the kind of details that are useful if you want to actually progress towards doing that. Otherwise, mostly, if the confidence is really high, then your X-ray experiment will end up verifying AlphaFold, right? Yeah. That you have to keep in mind.
I'll take questions, but let's first start with Ullas.
Yeah, Ravi.
Yeah.
So what is the error thresholding here? I mean, I'm just wondering, there should be an error filtering. So what is the level to which you're error filtering in this? And the second thing is because increasingly more proteins have been known to have unstructured domains and intrinsically disordered regions.
Absolutely.
So how does DeepMind or AlphaFold work in very structured domains versus unstructured? Is there any sense there?
Yeah, there is. So see, essentially, it's a little bit like the human mind, what he was talking about, right? If I know a sequence, let's say, for example, you as a biochemist have studied proteins or different sequences, for example, right? And you have a certain amount of experience. Like if you see a particular sequence, you know that this is going to be a disordered domain, for example, right? So AlphaFold is the same way, actually. It's seen enough and it has learned enough that basically when it sees a particular sequence or whatever it is, it's able to tell that this must be part of a disordered domain.
But you can't predict the disordered region.
But predicting the disordered regions actually has no, I mean, in some sense it has no meaning because the thing is disordered. So there is no, minimum structure, for example, or solid structure that it can actually put out and convince you that this is the structure, because that structure is going to fluctuate, right? It is very flexible, it's loopy, it's floppy. So what AlphaFold is telling you is this part is floppy. That's it. So now as a crystallographer, you go for actually determining the protein structure. AlphaFold tells you that this part is floppy. You put in crystal contacts to make sure that you stabilize that, and then you crystallize your protein. So maybe yesterday you were actually doing this crystallography, basically, and the crystals were not growing at all because the loops were floppy loops, right? They were not stabilizing, for example. You put the right contacts and do the right mutations, basically, now you can crystallize your protein, lo and behold, right? So that's the kind of information that's valuable in this case.
What's the next speaker of the question?
Yes.
So in much simpler applications of deep learning, often there's this phenomena that of instability. So like the celebrated examples are things like that you would just train a network to detect traffic signs, and it's a no board. You change a few pixels. To any human, it looks still like a stop board. If it's a stop board, it looks like a stop board. But the network, which is trained very well, doing very well, gives completely confidently an answer that it is a go board.
All right.
So I have those kind of studies being carried out with these, and like these, and with the experimental thing, like especially because all of those structures have never been experimented. There might be things like that lurking in that. Also, right?
No, absolutely. So if there are no, there are no structures around, and this is the only game in town at this point. So how much do you trust this particular structure actually? So I guess that's why you need,
especially in rare events.
That's why you need an extensive validation set, which will actually cover this actually, right? So the CASP competition is one of the most the fact that they entered the CASP competition with AI.
But
is laughable. I think at some level.
But what I'm saying is that like, even in those things, those models have been like all of these things have been done there. And in fact, to much more, because these problems are much simpler, so it's much easier to do all those things.
Right.
Still, you sometimes find that they're unstable.
No, that's true. So all those,
so it can be much more subtle in a more complicated model.
I couldn't agree with you more actually. But essentially that was one of the few things that they were careful enough to actually take care of these instability. So they brought in. Demis actually has the expertise in terms of the AI ML aspect actually, and John brought in not only the engineering but also the biophysics and the simulation background overall with respect to that. Yeah.
Hi, very nice talk. There's a quick question regarding the next steps on these areas. So, as deep learning is already able to predict what they should be seeing now, and experimentally one is able to validate that, are there situations where people are trying to change the physical parameters, say the temperature, or going deep in the ocean and the extreme atmosphere and scenarios of what kind of proteins one would expect, which has never been seen so far?
Yeah.
So are there such efforts going on, and what is the next step for deep learning?
So it's deep learning, right? So it depends on data. So if the data is sparse, right, there is no learning that's going to happen actually in that particular case. So this is a challenge. Like in the sense, if you want to go outside the conventional, like the standard thing that I was discussing with Ullas about intrinsically disordered protein systems, the data is just not there actually. So how is the network going to learn enough for you to give you reasonable things, for example? So yeah, that is still up in the air. But again, here another point basically where you need solid data. This was possible because there was solid experimental data. That elbow grease of 0.2 million structures was very, very important actually to get this. Yeah.
Okay.
Yeah.
Last two questions. Please help me in finishing this and we can walk out together with some facts. So there will be chance where you can talk to Ravi. So last two questions.
Yeah. So my question is, so certain proteins are easier to isolate, so they have better structures. So let's say it's a transmembrane protein. You will have less of them in the database in the very first place. So they will, when you use this to predict, so you will be making errors because the fundamental models have, the database had models which had errors or which were of very few in numbers. So you inherently have a systems bias. So were they trying to correct themselves? Was there a validating step?
Yeah. So all of them, I mean, like whatever structures they were predicted, so there is a training set, then there is a validation set, which is actually very important, and then finally there is the prediction towards things which are completely unknown.
So there is a training.
So it's actually a variant of what.
I know. So there is a training step, there is a test step, then there is a validation step also, right? Yeah. So and the second thing that I'm asking is, when you use AlphaFold, it spits out around like 5 structures, right? So there is a structure 1, 2, 3, 4, 5 based on a ranking, right? So are those the remaining 4 are local minima?
Yeah. So the physical. So essentially, those are all scored, right? So essentially, you'll have a structure with the best score, and so on and so forth, right? So now, what do those structures represent? So there have been hypotheses in the literature that those structures actually represent other structures on the minima, which are close by, for example, right? But there is no because this thing. So there is also a hypothesis that the program has learned an underlying statistical potential, which connects the sequence to the thing basically. But what is that thing that it has learned? We don't know how to pull that out. I mean, Piyush can tell us basically. So because that's present in some feature space in terms of the weights, etc., that is encoded inside the network. Can we interpret that to actually bring out what is the underlying landscape that it learned? It's up in the air actually. Nobody knows.
Okay. We thank everyone for coming, and just wanted to tell that John Jumper was a TMQ lecturer in February. So we had a great opportunity to talk to him and host him for a biophysics meeting.
Yeah, I just want to say basically, I had the privilege of meeting John Jumper, and I got into an argument with him about whether protein dynamics is important or not actually. Right? He kept saying structure. I said dynamics, and now it's my punishment to give this Nobel talk with John Jumper actually.
Okay. On that note, that's time again, everyone.